people believed that life arose spontaneously from dirt and from the flesh of dead animals. The ancient Greeks taught that insects came from decaying matter. And even as late as the 1800s, there were still a few scientists who assumed that rats materialized from old rags and maggots from rotting meat. This view of life has changed. Scientists now know that the basic units of all life, the cells, come from other cells. The cells of our own bodies arose from a single cell, the product of our parents, and their cells from their parents, forming an unbroken chain begun eons ago. Thus, from the lowliest creature to the most complex, life persists by reproducing itself in a timeless process called cell division. Like other living things, cells have a life cycle. During most of its life, a cell grows and carries out the functions for which it is programmed. In one-celled organisms, such as this paramecium, this means carrying out all of the life processes involved in feeding, movement, maintenance, and metabolism. In multi-celled organisms, it means carrying out some specialized function. These cells from a human salivary gland, for instance, are specialists in secretion. For most cells, the life cycle also involves a period of cell division, where the cell duplicates itself by splitting in two. This process of dividing underlies the growth, development, and maintenance of all multicellular creatures, including human beings.
The billions of cells that make up our bodies are all derived from dividing cells, starting with a single cell, the fertilized egg or zygote, formed by the union of sperm and egg. The thousands upon thousands of duplications that take place produce new cells which take on new identities. Some become muscle cells, others blood cells, and still others skin cells. While all cells in our body are produced by cell division, some, such as these nerve cells, stop dividing once they reach maturity. Other cells divide regularly as a normal state of affairs. These red blood cells, for example, have a lifetime of only about 3 months. They are constantly being replaced through cell division. Still other cells divide only under special circumstances, such as when we receive an injury. A broken bone, for instance, will result in a surge of cell division in the affected area, which will stop once the damaged tissue is replaced.
We may take the body's capacity to function, grow, and heal for granted, but these processes involve remarkably precise and orchestrated cellular events. To understand these events, it is important to know something about the structure of the cell itself. Most cells, aside from bacteria and blue-green algae, are composed of 3 major parts: the cell membrane, the cytoplasm, and the nucleus. The cell membrane is the delicate, thin covering that surrounds all cells. It encloses the contents of the cell and regulates the flow of incoming and outgoing materials. The cytoplasm refers to the fluid-like material enclosed by the membrane. Within this material are tiny structures called organelles, which perform all the functions necessary for the maintenance and growth of the cell. The nucleus is the all-important control center for the cell. Here lie the genetic instructions which determine the structure of the cell and tell it how to function within the organism. Under the microscope, the nucleus appears as a large sphere within the cytoplasm. It is enclosed by a double membrane, the nuclear envelope. When it is not dividing, the cell's nucleus is filled with a fine entanglement of material that can be stained with certain dyes. This is the chromatin. It consists of certain proteins and long fibrous molecules of deoxyribonucleic acid, or DNA. Within these molecules of DNA is the information about the characteristics of the organism. Every cell of the body, regardless of its particular function, contains an exact copy of this DNA, which in turn contains all the information necessary to run, build, and maintain every system of the body. Before a cell divides, its DNA is replicated, so that by the time division occurs, the nucleus contains two complete sets of DNA. During cell division, the two sets of DNA become condensed and, along with various proteins, are coiled and folded into dense rope-like structures which can be seen through the microscope. These are the chromosomes. Each species has a set number of chromosomes. Human cells, for instance, contain 46 chromosomes, while a common mosquito has 6. As packages of DNA, the chromosome's job is to carry the genetic information from the dividing parent cell into the two offspring, or daughter cells. Thus, the essence of cell division is the production of two cells whose DNA is identical to one another as well as to the parent cell.
We said that the life of a cell can be pictured as a cycle, with growth and functioning as one part, and actual division of the cell nucleus as the other. The first part is known as interphase, the second as mitosis. Interphase is a much longer period than mitosis. During this time, the cell carries out its normal functions, such as growth, production of chemicals, and maintenance. Interphase is also the time that the DNA molecules in the nucleus are exactly copied. Without this replication of the DNA blueprints, cell division would be impossible. Eventually, when all the cell machinery is ready, cell division, or mitosis, begins. Although mitosis is a continuous process, it is helpful to follow the progress of the division through 4 major phases. The first phase of mitosis is prophase. Here the DNA, which up until now has been in long, invisible threads, becomes condensed into thick, visible chromosomes. Each chromosome now consists of 2 long subunits, each identical, called chromatids. Each chromatid contains an identical molecule of DNA. As prophase continues, the chromosomes become even more condensed, and a contracted region of the chromosome is seen. This is the centromere, which holds the 2 chromatids together. While the chromosomes are becoming more visible, the nuclear envelope surrounding them breaks down. Waiting for the chromosomes in the cytoplasm is the spindle apparatus. The spindle apparatus has 2 poles with bundles of protein cylinders called microtubules stretched between them. The last part of prophase occurs as the chromosomes attach to these spindle fibers.
The second stage of mitosis, known as metaphase, begins as the attached chromosomes begin to line up in a row, exactly between the 2 poles. It is during this stage that the chromosomes are the most highly condensed. The next stage is anaphase. This begins when the centromeres split in 2. Now, the sister centromeres move apart on the spindle fibers, pulling their chromatids apart after them. Thus, 2 groups of chromatids, each chromatid representing half of an originally double chromosome, move toward opposite poles. The fourth and final stage of mitosis, known as telophase, begins when the 2 sets of chromatids reach their respective poles. During this time, the chromatids loosen up and the DNA unravels from its condensed condition. Once this occurs, a nuclear envelope reforms around each cluster of DNA, and the daughter nuclei separate. Each of the 2 nuclei now contains identical sets of chromosomes. Finally, the cell itself is ready to actually divide. In animal cells, this process, known as cytokinesis, occurs as the cell simply pinches itself in two, separating the cytoplasm into two new cells, each containing one of the daughter nuclei. In plant cells, cytokinesis also involves the formation of a cell wall between the two daughter nuclei. To recap then, the process of mitosis involves four stages: prophase, metaphase, anaphase, and telophase. Whether in a plant or animal cell, mitosis culminates in the process of cytokinesis, where a parent cell finally separates into two complete daughter cells.
While properly regulated and timed cell division underlies all of life, uncontrolled division of cells, otherwise known as cancer, is a major cause of debilitation and death in our country. Scientists have yet to discover the precise cause of cancer, but most believe it involves a breakdown of the cell's response to stimuli that normally stop cell division. For instance, one study shows that normal cells stop dividing when the glass dish they are in becomes too crowded. This phenomenon is known as contact inhibition. Cancer cells, however, continue to grow and divide without stopping, seemingly unaffected by contact with other cells. Many forms of cancer are now treated with powerful chemicals and radiation exposures, which are designed to slow down and stop the wild growth of cancer cells. Until more is learned about the fundamental causes of cancer, these treatments, although limited, appear to have the greatest chance for success.
We have seen how the process of mitosis, when strictly regulated, allows the cells of the body to duplicate themselves. This same process enables certain organisms and plants to replicate their entire structures, a phenomenon known as asexual reproduction. A common example of an organism which can reproduce asexually is the starfish. Through cell division, a single piece of a starfish will generate into a whole organism, genetically identical to the original. This process helps maintain a large population of starfish in conditions that are often harsh and violent. On land, some organisms, such as this fungus, reproduce asexually by forming spores, which are released from the undersurface of the cap. These spores then grow into new organisms, again, genetically identical to the original. Some flowering plants can reproduce asexually by runners. The plants at the end of the runners simply take root, becoming new plants, genetically identical to their parent.
Through asexual reproduction and cell division, we see that mitosis maintains the continuity of life by handing down to the next generation accurate, exact copies of DNA, and thus exact copies of a cell's genetic makeup. Yet for many organisms, including humans, reproduction is carried out by a different process, known as sexual reproduction. Only through sexual reproduction can individuals combine to create new offspring with a multitude of variations. These variations create the shapes and personalities of our own species and the different forms of the creatures around us. In Part 2, we will learn about sexual reproduction and the special cellular division that makes it all possible. Imagine a world in which everyone was identical, where all humans shared the same thoughts, physical appearance, afflictions, and personalities. In such a world, there could be no individual creativity, no variety of interests, no motivation to achieve. Indeed, it is doubtful that such a species could even survive. Fortunately, humans are rich in diversity. Although we are all members of the same species, no two of us look or act exactly the same. This uniqueness is due in part to our ability to sexually reproduce. Through the process of sexual reproduction, the unique genetic makeup of two separate individuals comes together, and a new, distinctly individual member of the species is born. In part 2 of this program, we will explore the microscopic events which underlie the process of sexual reproduction and the special form of cell division which makes it all possible.
The ability to reproduce sexually depends on the formation of special sex cells known as gametes. Male gametes are called sperm, female gametes eggs. Gametes are different from most other cells in the body because they contain only half the usual number of chromosomes. While the majority of human cells contain 46 chromosomes and are referred to as diploid, the gametes contain only 23 chromosomes and are called haploid. This distinction is vital since it underlies the processes of sexual reproduction. When two haploid cells unite, the resulting zygote, or fertilized egg, is diploid, having received half its chromosomes from its mother and half from its father. Obviously, the process of forming sex cells is crucial to all species that reproduce sexually. This special form of cell division is known as meiosis. In understanding meiosis, it is important to keep one thing in mind. Chromosomes exist in pairs. We said that the normal human diploid cell contains 46 chromosomes. A more accurate statement is that it contains 23 pairs of chromosomes. Each pair is referred to as a homologous pair, and each member of the pair a homologue. Both members of a homologous pair are the same size and shape, and both contain genes governing the same traits. For example, if one of the genes responsible for eye color were located on this section of one chromosome, its mate would be located on the same section of the homologue. Notice that while a gene for a certain characteristic, in this case eye color, is located on the same part of each homologue, it is possible for one homologue to contain information for blue eyes, while the other contains information for brown eyes. Through meiosis, homologous pairs are separated, and haploid sex cells containing unpaired chromosomes are formed. Keeping this in mind, let's take a closer look at what scientists have discovered about the process of meiosis itself. While there are similarities between meiosis and mitosis, meiosis is more complex because it is a two-step cycle containing not one, but two divisions. As with mitosis, meiosis depends upon the replication of DNA in the dividing cell. Each of the 46 chromosomes in a typical human cell is duplicated prior to cell division, and two-stranded chromosomes held together at the centromere are formed. As you may recall from Part 1, each strand of the doubled chromosome is referred to as a chromatid, and each chromatid contains the exact same genetic information. For the sake of clarity, we will follow the meiotic division of a cell with only 4 chromosomes. The sequence and nature of meiotic events, however, is the same for human cells.
The initial phase of the first division is prophase 1. Here, as in mitosis, the chromosomes first come into view as they coil and become shorter and thicker. But now a fascinating thing happens unique to meiosis. Each member of a homologous pair matches with its counterpart, and the pairs of chromosomes line up tightly next to one another. This coupling of homologous pairs is known as synapsis. Sometimes during synapsis, A chromatid of one homologue will exchange genes with a chromatid of its corresponding homologue. This important mechanism, crossing over, will be more fully explained later. At the end of prophase 1, the chromosomes are fully contracted and quite short. Also during this time, the membrane of the nucleus has broken down. The next stage is the first meiotic metaphase. Here, a spindle forms in the cell, and each united homologous pair, now called a tetrad, lines up in a row halfway between the spindle poles. The first anaphase of meiosis starts with the movement of the chromosomes to the poles. The two chromatids of each homologue stay together, but the homologues themselves are pulled towards opposite poles, thus splitting the tetrads in two. The first telophase begins as a nuclear membrane forms around the chromosomes. Each new daughter cell now contains half the number of chromosomes of the parent cell. Notice, however, that the chromosomes are still double-stranded, made up of two chromatids attached at the centromere. The first division is now complete. The very brief period of time between the first and second meiotic divisions, known as interphase 2, is different from the first interphase in a very significant way. The DNA of the cells does not replicate. The second prophase begins with chromosomes once again becoming thicker and shorter, and a spindle apparatus forming in each cell. Metaphase 2 occurs when the chromosomes line up between the two poles. At the second anaphase of meiosis, the centromeres divide and the chromatids, now called chromosomes, separate and move to the poles. Finally, in the second telophase, the chromosomes in each group uncoil and are surrounded by a nuclear membrane, and a new cell membrane forms around each new cell. The end result of meiosis, then, is the creation of 4 distinct haploid cells from a single diploid parent cell. Not only does each of these sex cells contain half the number of chromosomes as the parent cell, but the chromosomes themselves have been altered through the process of crossing over, which occurs during the first prophase of meiosis. Scientists have discovered that during this period of contact, chromatids actually exchange genes at the points of contact. In humans, for instance, a gene for brown hair might be exchanged for that of blonde hair, or a gene for blue eyes might be exchanged for that of brown eyes. In addition to crossing over, chromosomes are in a sense shuffled during the first anaphase, with maternal and paternal chromosomes moving randomly to either pole. This shuffling of chromosomes is referred to as random assortment. Random assortment of chromosomes makes it more likely that the gametes will receive a combination of both maternal and paternal chromosomes. Although it is possible that each cell would receive strictly maternal or paternal chromosomes, the odds against it happening are about 8 million to 1. It would be like flipping 23 coins simultaneously and having them all come up heads.
While all meiotic divisions involve the transformation of one diploid cell to 4 separate haploid cells, the timing of the process and the structure and function of the gametes produced differ between males and females. In men, for example,